Hello. Hello.
Yes, hello.
Okay, so you guys can hear me?
Yes,
Yes, okay. So I believe Dr. Hamza or Dr. Ahmad, they are busy somewhere, so we will start with biochemistry today, okay? And let me share my screen so we can discuss this, okay? So for biochemistry, I'll try to cover the all important points in 3 to 4 days, okay? And what we will do is we will focus on the high yield stuff. Also, I'll give you the concept that you should always know, okay, but there might be few things that could have been missed, but since we are short on time, so we can do it in this way, okay. So we will start with biochem first, okay, and then we will discuss the genetics. The genetics is going to take like 1 day or something, but like your biochem part, like the biochemistry part, it will take like 2 days or 2.5, okay. So we'll start first from this ethanol metabolism. What I really want you to understand for this ethanol metabolism is first thing that you should always keep in mind. Someone speaker is on. Like, can you mute yourself? Yeah. So So I was telling that in ethanol metabolism, what happens is the first thing that you should always keep in mind is focus on this NADH to NAD plus ratio. Okay, this is the most important thing that you should keep in mind. Whenever they talk about alcohol metabolism, why are you seeing lactic acidosis? Why are you seeing ketogenesis? Why are you seeing lipogenesis? And because of this lipogenesis, we know like you see hepatic steatosis, which is the common cause of your cirrhosis, correct? So all this stuff, this is because of this the problem in this ratio. This is the first point that you should always keep in mind, okay, the problem in the ratio, okay. And what is happening here is that your ethanol is basically converted into acetaldehyde, and this acetaldehyde is converted into acetate, okay. And all this thing, when it's converted, it's producing your NADH, okay. So if I want you to assume that your body, someone has drank alcohol, and now there these two enzymes will work, you can say, yeah. Hello. Yeah. So I was saying that in this, you produce your NADH. Okay. So what happens is that your body tries to metabolize that ethanol. Okay. To, you can say, less.
Yeah, we
Okay.
the rose syrup. Yeah,
Um,
ice. I want
it's a lot of noise.
large. This is
can we have the page
okay.
number that you are following?
Yeah, this one is okay. Yeah.
Can you see my screen, guys? You
Yeah.
can see my screen, I believe.
Yes.
Can you mute yourself? Like whoever is unmuted, please. Okay. So at this point, I was telling you that we metabolize the ethanol into acetaldehyde and acetate. These are your, you can say, inactive metabolite. Okay. And what we do is we are producing more and more NADH. Whenever your ethanol is metabolized, you can remember that what you produce is NADH and what you consume is NAD plus, and that's why your this ratio thing is increased, because your, you can say, numerator is increased and your denominator is decreased. That's what really happening, correct? And whenever you have a lot of NADH, this literally means that you have high energy stores. And when you have high energy stores, what you do, you do not want gluconeogenesis, you do not want more TCA cycle to be working on, right? And you do not want, you can say, glycogenolysis, you do not want this. What you do instead, you make more and more lipids from that. That's why what pathways are, you can say, directed when you have high levels of NADH, your pathways towards your ketone productions or your lipid production. Okay, that's the main thing that you should keep in mind. So first thing, we focused on the ratio. NADH is always increased compared to NAD plus. In ethanol metabolism, this is going to be tested a lot on your exam, in your assessments, in your real exam. It's like very high yield concept that you should always keep in mind. And because of this whole thing, you see that your TCA cycle, your gluconeogenesis, they all are inhibited. Okay, and what happens is really that your increased ethanol metabolism that produces high NADH. Okay, so what happens is it inhibits your gluconeogenesis by, like, in what way they can ask you, they can inhibit your gluconeogenesis by decreasing the conversion of your malate into oxaloacetate. And if you cannot convert your malate into oxaloacetate, you cannot convert your oxaloacetate into phosphoenol pyruvate. This I'll come to in next slide. We will talk about this gluconeogenesis and glycolysis. For now, you can just know that you, in your gluconeogenesis, what you actually do is you convert your from your TCA cycle, you use protein or amino acid as a substrate to produce your glucose. This is your gluconeogenesis. And what literally happens is that in your TCA cycle, you convert your oxaloacetate, it's your amino acid, into your phosphoenol pyruvate. Okay, and this is the main thing that is happening here. This is inhibited, and you cannot convert in the forward way, like from oxaloacetate into phosphoenol pyruvate. Okay, and what happens is this is in an attempt to restore this NAD plus levels. Okay, that's the only attempt the body is trying to do, to compensate for the high NADH thing. Okay, so it inhibits this, and that's why you see what kind of symptom you can see. You can see fasting hypoglycemia. Okay, the other thing that you need to focus on is you do not undergo TCA cycle. And the so yeah, another point I wanted to make here is your pyruvate is basically converted into lactate, because conversion of pyruvate into lactate again generates your NAD plastic. Okay, so that's why two things are happening. First, your gluconeogenesis is inhibited. Okay, and why? Because you want to compensate for high levels of NADH, so you produce more and more NAD plus. Okay, and your pyruvate is converted into lactate because you want to produce more and more NAD compared to NADH. I hope this is very clear to you guys. Okay, and it means you have excessive acetyl CoA. And where should it go? Because it cannot undergo TCA cycle, it cannot be converted into oxaloacetate to go into gluconeogenesis. So where does it go? This acetyl CoA, excessive acetyl CoA, is then directed towards your lipogenesis. Okay, so that's why you see hepatosteatosis. Okay, that's why you see hepatic steatosis. This is the first thing. Another thing is that during your you can say glycolysis, what is happening is you have some intermediates which we will just discuss in a second. But one of your glycolysis intermediate is your glycerol aldehyde 3 phosphate, which is converted into DHAP, and this DHAP is converted into glycerol 3 phosphate. And what it actually produces, again, it restores your NADP plus. Okay, so this is the main thing that is happening here. Okay, this is the main thing that is happening here. So I believe this whole thing is very clear to you guys. Ethanol metabolism, what is happening, increase NADH compared to NAD plus, and that's why your all pathways that produce NADP, NAD plus, they are you can say stimulated. And one of the pathways is your oxaloacetate conversion into malate that produces your NAD plus. Another is your DHAP converted into glycerol 3 phosphate, which also produces your NAD plus, and that's why it helps with lipogenesis. And this can present as high fasting hypoglycemia, and lactic acidosis is always there whenever you consume ethanol. Why? Because your body wants to compensate by producing more and more NAD plus. Okay, so I believe we're very clear on this. I'll go fast with this biochem sessions because they are more of, you can just listen to me and follow me and you will be very good to go. Okay, and then you can just give it a read if you want. But that's these are the main points that you have to keep in mind. Another thing I wanted to tell you guys is that your ethanol is converted into acetaldehyde and this acetate stuff. This is more, you can say, common in men. Okay, and females, they do not have high levels of alcohol dehydrogenase. Okay, and that's why what happens is that females, you can say, if one glass of your alcohol can lead to a lot of effects in female in comparison to males. Why? Because they do not have this enzyme, so they do not metabolize the ethanol uh, in much quantity compared to males. Okay, so that's why they can suffer. That's why you can say that females are more susceptible than males because of low levels of activity of this alcohol dehydrogenase. It means in their bloodstream the ethanol will circulate in its whole form. It is not metabolized into something. Okay, but in males it is there, so it helps with the metabolization. And of course body size and body water and all this also plays a role. So I believe we are clear on this and we can move forward. This is just fomepizole is a competitive inhibitor of alcohol dehydrogenase, and your disulfiram is inhibitor of acetyl dehydrogenase. Less likely that you will get some questions on this, but for ethanol toxicities, always these are the things that these two drugs you should always remember and what they do. Just simple as that. Okay. And this fomepizole is also used for your ethanol toxicity, for your ethylene glycol or methanol toxicity as well. Okay, so these toxicities are also treated by fomepizole. So this is all about ethanol metabolism. Then we will now discuss this is actually a good review chart. The main thing here that you need to consider is that all these pathways, okay, all these pathways lead to one center point. This is your acetyl CoA. This acetyl CoA can be converted into it can go into TCA cycle to produce energy. It can go into gluconeogenesis. This acetyl CoA can go into lipid metabolism, like everything. So this is your center, you can say it's your center of attraction in all these biochem reactions. Okay, and we will discuss all these, but first I want to make sure that we discuss those topics, then these review charts will make sense to you. So 2 points here, two things here we will discuss today because we are going to discuss them, is your glycolysis and gluconeogenesis. Okay, so I want you to consider in your glycolysis what you are actually doing is you're trapping the glucose inside the cell and utilizing it and converting it into the form of acetyl CoA, and then this acetyl CoA can go into TCA cycle and lead to the energy production and all this stuff. Okay, and then it goes to electron transport chain where you have oxidative phosphorylation for your energy production and ATP production. Okay, this is the whole thing that is going on. So first you need to trap the glucose and convert it into acetyl CoA. This is your first step for your glycolysis. All these reactions that are in the mid, they do not carry much value. First thing for exam at least. Another thing, they are bidirectional and they can be easily converted into like this 3 phosphoglycerate can be easily converted into 2 phosphoglycerate. You do not need different enzymes, so these are bidirectional. They can be easily converted into one thing or other, like they you can say they can be exchanged into one another very easily. Okay, but what is of unique significance here is that your 3 steps, they, you can say they are bidirectional, but they need special enzymes, okay, to be bidirectional, otherwise they are unidirectional. Okay, by this I mean like glucose, it can be converted into glucose 6 phosphate by hexokinase or glucokinase. This we will also talk about, like why we have these two kinds of hexokinase and glucokinases. But for simplicity purpose and now, just stick to this, that your glucose is trapped inside the cell by the utilization of your enzyme hexokinase or glucokinase and converted into glucose 6 phosphate. Okay, but when you suppose that you are undergoing your gluconeogenesis, right, then you convert this glucose 6 phosphate into glucose. right? You provide for the energy in gluconeogenesis because it's neo, it's like new glucose production. So when you provide for the glucose to the peripheral, you can say body, right? You need to free up your glucose from that cell, like liver cell, to your peripheral tissues, right? So what happens is your glucose 6 phosphate is converted into glucose by a different enzyme, that is glucose 6 phosphatase. Okay, so this means that I can undergo glycolysis easily, but when I need to go the reverse, that is your gluconeogenesis, at 3 steps I would have to use different enzymes. And if for suppose those enzymes are deficient or there is some kind of problem with those enzymes, I cannot produce your glucose when the person is fasting, especially prolonged fasting. When the glycogen storage is depleted, then body relies on gluconeogenesis. It means at that point I cannot, you can say, utilize glucose, correct? Because my that enzyme is missing, and that enzyme here is your glucose 6 phosphatase. Same goes for your the other 2 steps that I'm going to tell you. First is your, okay, so first is your fructose 6 phosphate converted into fructose 1 6 bisphosphate. Okay, when you go into the glycolysis, the enzyme that helps with the conversion is your phosphofructokinase. And how can you remember this? Just remember that kinases are the enzyme that attach the phosphate. Okay, these kinases are the enzyme that attach the phosphate. Okay, and your bisphosphatase, like phosphatase, like whatever ends with tase, is the enzyme that removes phosphate. Okay, that removes phosphate. These are the important points that you should keep in mind, and it will help you, like what is, at least you will get some idea what is happening inside the body. And so yeah, so you are at this point, your fructose 6 phosphate is converted into fructose 1 6 bisphosphate. Okay, by the enzyme phosphofructokinase 1. Okay, by the enzyme phosphofructokinase 1. And if you want to go the reverse, it means you converted in your oxaloacetate into phosphoenol pyruvate, and now you are undergoing gluconeogenesis. If you reach this point, if you made fructose 1 6 bisphosphate, you cannot convert this into fructose 6 phosphate by the same enzyme. You need different enzyme, and that different enzyme is your bisphosphatase, because it's removing the phosphate. So it's fructose 1 6 bisphosphatase 1. Simple as that. Okay, and then what is happening is, this is your second step, that is bidirectional, but technically it's again unidirectional because you need different enzymes. Okay, and your third step, that is also like similar to this, is your the conversion of your phosphoenol pyruvate into pyruvate. Okay, this is again unidirectional. What happens is you convert your phosphoenol pyruvate, okay, into your this pyruvate by your pyruvate kinase. Okay, because you are again attaching a phosphate here, and you can say controlling your conversions. Okay, so again by pyruvate kinase you convert your phosphoenol pyruvate to pyruvate. Okay, and from pyruvate you go undergo into TCA cycle either by acetyl CoA or either by oxaloacetate. If you convert your pyruvate into oxaloacetate, you utilize the enzyme pyruvate carboxylase. Okay, you can remember it's oxaloacetate, so oxylase, carboxylase, like carboxylase. So this word is very similar, oxaloacetate. So okay, so maybe it helps you remember that pyruvate is converted into oxaloacetate by carboxylase. Okay, so after this, your oxaloacetate, and now this is the thing, you utilized your glucose, you converted it to phosphoenol pyruvate, then it converted into pyruvate, and now it's converted into oxaloacetate. Okay, and you underwent TCA cycle. But now you are in fasting state. It means this oxaloacetate needs to be converted into pyruvate. But there's a trick, that your body does not convert directly oxaloacetate into pyruvate. Instead, it converts your oxaloacetate into phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxykinase, like PEP carboxykinase. Okay, so oxaloacetate is basically converted into PEP by PEP carboxykinase. Okay, and then again it goes up and up until this point. Then you utilize a different enzyme. Then again at this point, then you utilize again a different enzyme. So I believe this whole thing is very clear to you. These are very, you can say, you do not need to know everything for biochem on your exam. You need to know the bullet points. And this is your bullet point for your glycolysis and gluconeogenesis. Okay. So I believe this whole stuff is very clear to you. And I would like to say that okay, so this will be covered in vitamins. So yeah, this is it about okay. And before we move on to this, I'll want to talk about this, okay. So this is something that they like to go after and they like to test because it's very volatile, it's very similar, but I'll try to make it very easy for you, like as much as I can. And in this you have to remember, like, now focus on this. I told you your fructose 6 phosphate is converted into fructose 1,6 bisphosphate, okay. And then the whole reaction goes down, correct. And because this step is actually the rate limiting step, okay. So what happens is this is tightly regulated, you can say. So this is your fructose 6 phosphate that is converted into fructose 1,6 bisphosphate and then goes into glycolysis, the same as the previous one. I hope this you guys are getting this right. And then fructose 6 phosphate, if I want that this fructose 6 phosphate should undergo gluconeogenesis. It means my fructose 6 phosphate should be converted into fructose like glucose 6 phosphate, and then from that it should be converted into glucose and undergo gluconeogenesis, right. So here I want you to focus on these words. Whenever you see kinase, whenever you see kinase, okay, I want you to keep this in mind that you will, whenever you see kinase, it means you will attach the phosphate. So you will convert your single number like 6 to 1,6 or 2,6. Okay. Whenever you use kinase, you will either convert it into 1,6 or 2,6. If you use phosphofructokinase 1, you will make your fructose 1,6 bisphosphate. If you use your phosphofructokinase 2, you will make your fructose 2,6 bisphosphate. And whenever you will have higher two numbers, right? Whenever your kinase is activated, or you can say you will have higher number of these things, like 1,6 bisphosphate or 2,6 bisphosphate, you would like to go glycolysis. That's the main thing. So kinase makes your two numbers. You can say, you can remember it in this way, that kinase makes two numbers, okay. And if I have high two numbers, I'll undergo glycolysis. That's the main thing that you should keep in mind for easiness and for memorization, okay. And when you go reverse, like you want to go reverse and you want to utilize your glucose in the periphery or not in the cell, what, and you like you are going undergoing gluconeogenesis, what you actually do, you utilize phosphatase, like a bisphosphatase, okay. Fructose bisphosphatase 1 or fructose bisphosphatase 2, okay. And whenever you use phosphatase, it means you are reducing the number, you are making it one number thing, okay. And if you make it lesser number, it means you are it means you are in fasting state, correct. And that's why you will undergo gluconeogenesis. In this way you can remember it, okay. So I believe this is all clear to you guys. If you are confused, you can just let me know before I proceed. Just anyone can just say that, like, it's very clear to them.
Doctor, could you please explain again the kinase and phosphatase? Just the
Yeah,
last
sure, sure,
line.
sure. Okay. Yeah, yeah, okay. So I was saying that first, understand this kinase always means attaching phosphate. Okay, attaching phosphate. And whenever your body is high in, you can say, energy, then it wants to attach the phosphate, because phosphate is actually the, you can say, ion or element that provides the energy, right. So it means you are full in your fed state. That's why you want to undergo glycolysis. You do not want to undergo gluconeogenesis, right. And that's why this kinase will attach phosphate to fructose 6 phosphate and either make it 1,6 phosphate or 2,6 phosphate. It really depends, right. And I'm going to tell you first, try to stick to this concept, okay. So 1,6 phosphate or 2,6 phosphate, and if you have two numbers, it means you are having higher energy, it means you are undergoing glycolysis. So whenever you convert your fructose 6 phosphate by using kinase enzyme, it means you are producing two numbers, either fructose 1,6 bisphosphate or fructose 2,6 bisphosphate, okay. And it means you are undergoing glycolysis. And reverse is also the same. Whenever you use phosphatase, Okay, whenever you use phosphatase means removing the phosphate. It means I remove one phosphate from this fructose. It means I remove this phosphate which was at position 2, and here I remove phosphate which was at position 1, right? And simply will make it fructose 6 phosphate, correct? Because that's the logical mathematics you can think of. So whenever my kinase is activated, I will undergo glycolysis because I will attach more and more phosphate. Okay. Whenever my bisphosphatase is activated, that means I am low in energy stores and I'll remove the phosphate and I'll undergo gluconeogenesis. Correct? Okay.
Thank you so much. Got it.
And after this, yeah, yeah, no worries. So after this you can just keep in mind upon other point. Yeah, so like what is the reason we did not study this step in your glycolysis, right? We studied just this, like fructose 6 phosphate is converted into fructose 1 6 bisphosphate, right? So these two enzymes are basically your regulators, you can say. These are regulating the enzyme, the activity of this phosphofructokinase 1 and bisphosphatase 1. That's the main thing. So you can say this, that this reaction, conversion of fructose 6 phosphate into fructose 1 6 bisphosphatase, right, is regulated by these two enzymes. And these two enzymes, how are they regulated? It really depends upon your body glucose stores. If you are in fed state, your kinase will be activated. If in your fasting state, your bisphosphatase will be activated. Simple as that. Okay, so I believe this is very clear to you guys, right? You can find some few questions on this. They can just ask you this. Don't confuse with the names. Try to understand the terminology, as I have, like, I believe I have made it very easy for you. So in this way you can just keep this in mind. Okay. And other point I will make, I told you, like, these two enzymes are regulating this whole pathway, correct? And what is their end product? This their end product is fructose 2,6 bisphosphatase, right? So again the same thing, like 2,6 bisphosphatase will try to activate and produce two numbers, so fructose 1,6 bisphosphate. Same as that. Okay, so I believe this is very clear to you. I'll just read it out so that you are not confused. So whenever you are in fasting state, your glucagon is high. It means you will undergo gluconeogenesis. So what should be high? Your bisphosphatase. And what should be low? Kinase. Okay. And this means less glycolysis, more gluconeogenesis. Okay. This point I'll just come to it in next few minutes. Okay. And fed state, it means your energy stores are high. So what should be happening? Your kinase should be high, because I told you kinase is the one that attaches the phosphate, and it really means you are high in energy stores. And your bisphosphatase should be down, correct? Because you're having high energy stores. More glycolysis, less gluconeogenesis. Okay. Just two more points that you have to memorize, and they can be asked. So just keep in mind that fasting state, glucagon is high. Fed state, insulin is high. Whenever your glucagon is high, you need to remember your two things are also high in this pathway: your cyclic AMP, okay, and your protein kinase A. There is no logical sense that I could make for you to memorize this, at least, because these are just reactions. They are just there because they are there, and there is no logical thing to make it fit and memorize it. But to make it easy, if you follow what I told you after this, you can just remember that whenever I'm fasting, I will have high levels of both the things: cyclic AMP and this protein kinase A. And whenever I'm in fed state, I will have low level of these both things. Simple as that. Okay, for simplicity purposes. And they can literally ask you, like, what happens when you are in fed state, what happens to your protein kinase A, what happens to your phosphofructokinase 2, or phosphofructokinase 1, things like this. Okay, so if you are all clear, so I can move ahead. Someone can just confirm it so we can move ahead. Hello.
Yes, doctor. Thank you.
Yeah, okay. Yeah, okay. So now there are few questions on this hexokinase and glucokinase. As I just told you, that you trap your glucose and convert it into glucose 6 phosphatase, right? By using these enzymes. So how are they different? This is something that you should always know for exam purposes, because this can be tested in the form of graphs or something. So the main thing here that you have to focus on is that hexokinase is present. Like this enzyme traps your glucose and converts it into glucose 6 phosphate in all cells of your body, except for your pancreatic cells and liver cells. Okay, and that's why in your liver cells and beta pancreatic cells you have glucokinase, not hexokinase. Okay, both do the same thing. The only difference is depending upon it's their purpose, you can say. Okay, so what is happening here is that first you need to understand Km means um, substrate concentration. Okay, like here what is our substrate? Glucose. Okay, and Vmax means you can say Vmax is like I can say this, like these enzymes, glucokinase or hexokinase, how fast or slow they are when they are partly saturated. This is your Vmax. Okay, so Vmax is a, you can say it tells that is enzyme fast or slow upon the depending upon the substrate, right? When this enzyme, you can say, is partially filled, like there are 100 hexokinases, okay, and if the 50 hexokinases are filled, how fast are they, or how slow are they? This is the main thing that you should keep in mind. Okay, and now if I say there's a thing like, now if you focus on these words, what they literally mean, it can logically make sense. How? If I say that your hexokinase, okay, it has low Km, it means whenever your body has, you can say, 70 units of glucose, suppose, in your body. Okay, and which enzyme would be more active? It's your hexokinase, not your glucokinase. Why? It means even at low substration, even at low concentrations, it has ability to bind with the glucose. This is your hexokinase. Okay, and this is logically also true, because all cells of your body want to trap the glucose inside them so that they can produce the energy. It means whenever you are on the lower levels of your glucose, your hexokinase is there to save your cells from dying, right? But for beta pancreatic cells or liver cells, it's not like this, because liver cells only store the glucose, you can say, and of course utilize some of that, but most of the it goes into storage in the form of glycogen and stuff. And for beta cells, we know glucose is picked up in order to release the insulin, right? So it means my glucokinase should only be present on these cells because it has low affinity. It means it has higher concentration, or Km. It means whenever I have high glucose levels like 200 or something, only then my insulin will be released. And this logically makes sense, because only then your cell wants to release your insulin. Because in this condition, if you release the insulin, it's leading to worsening of your hypoglycemia, right? So most of your bodies have hexokinase because they want to protect you from, you can say, starvation. They want to protect you from worsening of your hypoglycemia. And your glucokinase is only on beta pancreatic cells or on your liver cells, because they are there to regulate the insulin production when glucose is high in amount. That's the main idea here. Okay, so I believe this is very clear to you guys. And this means Km, as I told you, whenever you read the word Km, just say concentration. So concentration of glucose is lower, it means my hexokinase will be active, it will be working very well. And when my Km, it means concentration of glucose is higher, my glucokinase would be activated. And why? Because it wants to release the insulin. Okay, now if we talk about the Vmax. Okay, so for me, Vmax, you need to remember that uh, like you constantly need energy, right? And you constantly want to utilize and make energy, right? So if your body cells, all your body cells, if they rapidly metabolize your glucose, right, it is going to hurt you. because then you would want to eat again and again. This is not good for your body, right? So your Vmax is lower for hexokinase because it's present on all cells of your body. It means this enzyme has a basal activity. It maintains your basal glucose levels. It provides you basal energy levels because it utilizes glucose very slowly. But your glucokinase, it has higher Vmax. It means it utilizes glucose very fastly because here you have to be very active to release insulin because you cannot tolerate high levels of glucose continuously. That's the main idea here. So glucokinase is there to regulate your insulin production from your beta pancreatic cells. This is the main thing, and that's why glucokinase is unique. It has higher Km and higher Vmax. Okay, hexokinase is present in all cells of your body because it wants to maintain the basal glucose levels and do not want to worsen your hypoglycemia or whenever you have low glucose levels. Okay, and that's why it has lower Km and lower Vmax. That's the main thing that you should keep in mind. Okay, and I have seen like they can give you graphs like this is Vmax and this is Km. Okay, so they can give you what kind of you can say. It means they can give you something like this actually, right? It means this is higher Vmax, so what comes in your mind, what kind of enzyme it is? It's your glucokinase, right? Because it has higher Vmax, it comes here. But this has a half Vmax, it means this is your hexokinase. Okay, and same goes for glucose concentrations. Okay, so this is just a thing that you should keep in mind, just a concept. If you remember the concept, you will be able to solve any question whenever it shows up regarding Vmax and Km and glucokinase and hexokinase. Okay, so after this, um if someone did not get it, like I can repeat it. So if you guys get it, I can move forward. Just someone can confirm me because I have to go back to see your thumbs up. Okay. Pyruvate dehydrogenase complex, for this you just need to remember that the main thing here, if I were to tell you, it converts your pyruvate into your acetyl CoA. Okay, and what is unique about this pyruvate dehydrogenase complex is that it is composed of three enzymes. Like this itself is a enzyme, but you can think of, you can visualize it as a bigger enzyme that is composed of three smaller enzymes, and it also has cofactors, and these are your five cofactors: thiamine, lipoic acid, CoA, FAD, NAD. This can sometimes be asked. We will come to vitamins. When we review the vitamins, vitamins are very high yield for your exam. Like I'll tell you, for now you can just remember that CoA is B5 and FAD is B2, NADP plus is your B3 if you are learning for the first time. But if you know it, so it's okay. This is the main thing that you should remember for this. And of course, as I just told you, it helps with the conversion of your pyruvate into acetyl CoA. The similar, you can say, structural composition is also found in another enzyme, that is your alpha ketoglutarate dehydrogenase. Okay, and this alpha ketoglutarate dehydrogenase basically is a TCA enzyme. It converts your alpha ketoglutarate into succinyl CoA. So they can just give you some question like this, like pyruvate dehydrogenase is very similar to what kind of enzyme in your TCA cycle, something like this. Or they can ask you about the arsenic poisoning, and they can ask you if your lipoic acid, like basically your arsenic basically inhabits your lipoic acid. Okay, and if it inhabits your lipoic acid, what two enzymes can go down? It's your pyruvate dehydrogenase and your alpha ketoglutarate dehydrogenase. Okay, this is the main thing. And how does this arsenic poisoning actually present? It presents with pigmentary changes. This you should always remember, like vampire skin, and skin cancer can be there. You can see vomiting, non-specific. Garlic breath is very specific for this. Long QT is also tested. So long QT, garlic breath, and vampire skin. This is the main thing that you should always keep in mind for arsenic poisoning. Okay, and for pyruvate, this is very important. This whole thing here is very important. Okay, and for this you need to keep in mind that your pyruvate is basically converted into lactate, alanine, oxaloacetate, and acetyl CoA. It's the same thing that we discussed here. Okay, your pyruvate, lactate, alanine, acetyl CoA, and oxaloacetate, same thing. Okay, but in more detail, and what are the regulators and all this stuff. So the main thing that you have to keep here in mind, that conversion of your pyruvate into alanine again is bidirectional. Okay, and same goes for lactate, it's bidirectional. You do not need something else, you utilize only one enzyme, that is your LDH here, and your ALT here. Okay, so conversion of pyruvate into alanine is by ALT, alanine aminotransferase. Conversion of pyruvate into lactate is by lactate dehydrogenase. Conversion of your pyruvate into oxaloacetate is unidirectional. Okay, it's by pyruvate carboxylase. Conversion of pyruvate into acetyl CoA again is unidirectional, it's by your pyruvate dehydrogenase, like this enzyme. Okay, it's a bigger complex, you have to remember this. So these things you have to remember. What happened is your alanine aminotransferase, very important to keep in mind. Pyruvate converted into alanine utilizes, like these enzyme utilize your B6. Okay, this you have to keep in mind. They like to test vitamins as a cofactor, so very important, keep this in mind. Alanine, like ALT, I mean, utilizes your B6 as a cofactor. LDH, B3. Okay, pyruvate carboxylase, whatever enzyme ends with the word carboxylase, actually transfers your CO2 molecule to the product, to the end product. Okay, so it basically transfers your carbon from this to this. Okay, so it adds on a new carbon molecule, you can think of it this way. Okay, and if pyruvate is converted into oxaloacetate by using a carboxylase enzyme. Okay, so CO2. And what vitamin helps with this? B7. B7 is biotin. It always provides your carbon components, and that's why it's always a constant cofactor for all of your carboxylase enzymes. Okay, so pyruvate into oxaloacetate, and then pyruvate is converted into acetyl CoA by pyruvate dehydrogenase, as I just told you. And this again utilizes these all cofactors, I have told you about this. And lactate dehydrogenase is a form, like, why would you like to convert pyruvate into lactate? Because whenever you undergo anaerobic glycolysis, right, whenever you have low oxygen in your body, or you have anemia or something, or you're exercising very vigorously, then you want to undergo anaerobic glycolysis. So that's why it's major pathway in your kidney medulla, because it's a highly active, you can say, area of your body. Lens, cornea, Sertoli cells, WBCs and RBCs. RBCs are basically anucleated, so they can never undergo TCA and electron transport chain, because that requires your mitochondria. Okay, so that's why your RBCs rely only on your anaerobic glycolysis. Okay, and that's why RBCs basically produce less energy compared to all cells of your body. Okay, pyruvate into lactate, RBCs, main pathway for the energy production. And for this pyruvate into alanine, what it really does is carries your amino group from your muscle to the liver. Okay, when you have high intensity exercise, you produce your, you can say, amino groups, like you have this protein catabolism or ammonia production, stuff like this. It is picked up from your muscle to the liver, and where it is converted back into pyruvate. We will talk about this in more detail, we have it with the urea cycle, so don't worry about this. Okay, and we are done with this one also. For pyruvate carboxylase, as I've just told you, it is converted into oxaloacetate. It can go into TCA cycle, and of course, if you want your oxaloacetate to be converted into gluconeogenesis, you will convert it into PEP by PEP carboxykinase. You can never convert your oxaloacetate directly into pyruvate kinase. These are the main things that you should keep in mind, especially the cofactors like vitamins and stuff. Okay, and then this is not important as such, only one enzyme that I just discussed with you that you should keep in mind, they have written it here, but nothing specific, like very unlikely that you will see a lot of questions on this. The main things I'm covering them for you. Okay, for your etc, you should remember it's in your mitochondrial membrane. So this is your mitochondrial matrix, and this is your inner mitochondrial membrane, this is your interspace and all this stuff. The main thing here that you need to know is, in your mitochondrial membrane you have these protein complexes: complex 1, complex 2, complex 3, complex 4, and complex 5. Complex 5 is your ATP synthase that actually produce your hydrogen, like that actually brings back your hydrogen from your extracellular space into the mitochondria. Okay, and this pumping produces your energy. Okay, that's the whole idea here. And your all other complexes, they push or flush your proton into your cytosol or extracellular space, not into mitochondrial space or inner mitochondrial matrix. It's only complex 5. Okay. And that the why it is like this, because it creates a gradient. If you keep on pushing something here, it means there will be excessive amount of that thing, and now it's very easy for you to push it from this space to this space, because here the concentration is high and here it's low. Okay. That's the main idea here. The main thing that they can like ask, or they love to ask, is the clinical thing. They can ask you, how does the aspirin lead to metabolic acidosis, because it inhabits your this etc chain. Okay. And this actually inhabits your oxidative phosphorylation. It means you cannot pump your hydrogen ions into your extracellular space, and this also means you cannot pump it back to the mitochondrial space. So in nutshell, aspirin is very, you can say, bad drug. It inhabits the whole thing. Okay, the whole pump and the whole thing. And for complex 1, 2 and 3 and 4, what is unique is that they can be like your complex 4 can be inhabited by your cyanide and carbon monoxide poisoning. This can sometimes be tested, remember it. It's simple as that. They can just literally ask you, they will give you a whole scenario on cyanide poisoning or carbon monoxide poisoning and blue skin and all this stuff. And they will give you the oxygen saturation is low and everything else. And they can ask you what complex is inhabited, so you know it's complex 4. Okay. Or they can just give you cytochrome C and names like these. Okay. They can ask you about the succinate dehydrogenase. Okay. That is very unique, because all other complexes, they utilize your NAD. This is unique, and this is smaller complex as well. It's succinate dehydrogenase complex 2. This gives or provides energy by utilizing FADH2 and convert it into FAD, and in this way it provides for the energy. Okay. And this FADH2 and FAD is basically your one of your key, you can say, pathway in your TCA cycle. Okay. So here if you see, your succinate is converted into fumarate and makes your FADH2, and same thing happens here. Your succinate dehydrogenase makes your FADH2 and makes fumarate. So they can give you a question that some kind of a child is undergoing some disease and all this stuff, and they can ask you that the production of FADH2 is down. What kind of enzyme is inhibited? It's your succinate dehydrogenase. What kind of pathway is inhibited? TCA cycle. What end product you cannot form? It's your fumarate. What kind of energy you cannot provide? FADH2. Like the same thing tested in different ways. Okay. This you should keep in mind, and that's all for this one. Gluconeogenesis we have just discussed. This they have also talked about these key regulator enzymes that I just told you. So I'm not going through this because we have to be very quick. Also, the main thing here I have told you all. Just one more thing, that you are that you should always remember, your gluconeogenesis is done by liver cells and also kidney cells. This is something that they love to go after, testing that your kidney cells are also like your, you can say, contributing to your gluconeogenesis. It's very unique. And your GI tract is also, like your GI lining also can contribute to gluconeogenesis. So liver, kidneys and GI lining. Okay. And other thing is that whenever we say gluconeogenesis, it means production of your glucose from non glucose sources or non carbohydrate sources like amino acids like oxaloacetate. Okay. Or your fat component. By fat I mean odd chain fatty acids. Okay. So it's only the odd chain fatty acids that can be converted into your gluconeogenesis pathway. Why? Because in your gluconeogenesis pathway you can convert this odd chain fatty acid, which is basically your propionyl CoA. This you can just remember, it's a constant thing. Why it's odd chain? Because it's 3C, it's 3 carbon component. Your, for suppose acetyl CoA, it's your 2 carbon component, right? And because it's 2 carbon component, it can easily undergo TCA cycle, it or into gluconeogenesis, right? And all your fats that are broken into odd numbers can end up being propionyl CoA. all your fatty acids that are divided evenly, like in two carbon components, right? They can never end up being propionyl CoA. So it's only going to be your odd chain fatty acids that produces your odd number. That's why it's known as odd chain fatty acid. Okay, this you have to keep in mind. It's your propionyl CoA always, and this propionyl CoA can enter your TCA cycle as succinyl CoA. This you have to remember, like this state. Okay, your propionyl CoA can be converted into succinyl CoA. Okay, and after this, it means you will make oxaloacetate, and this oxaloacetate again will go into PEP, and you can contribute to gluconeogenesis. So that's the main thing. Only your odd chain fatty acids can contribute to gluconeogenesis. Okay, that's the main thing. Even chain fatty acids cannot produce new glucose, as I just told you, because they are even in numbers. So it means whatever end product you get by the metabolization of your fats, it can easily be converted into acetyl CoA. Okay, that's the main thing, because it's two carbon component, it can easily go here and there. Okay, so this pentose phosphate pathway, for this you need to keep in mind that what is the purpose of this pathway. This pathway is the main pathway for your RBCs. Okay, for your RBCs especially, because you can say it provides, it keeps your RBCs protected from antioxidants. How it produces NADPH. Okay, this pentose phosphate pathway, or HMP shunt, produces your NADPH, which protects your RBCs from your antioxidants, right? That's the main thing. And if I were to say your this is your RBC, like this whole thing is undergoing into your RBC, and here you have glutathione enzyme. Okay, you want to keep this glutathione reduced. If this is reduced, it means you are not going to damage your RBC. Okay, and for that, if you see, you require an ADPH. Okay, if for suppose you have high levels of this and low levels of this, it means this glutathione cannot be reduced. It will stay in this oxidized state, and if this stays in oxidized state, it can actually lead to this disease, glucose 6 phosphate dehydrogenase deficiency, or it can just lead to hemolysis, stuff like this. So your RBCs, they want to be protected by this antioxidant stress, and how they protect themselves by the production of this NADPH. And what they do is they trap the glucose, they can convert the glucose into glucose 6 phosphate, and then it goes into glucose 6 phosphogluconate, ribulose 5 phosphate. Do you really need to know all these steps? No. You just need to know the main enzymes like this and this. Okay, that is the main thing that you should you should keep in mind. Okay, this is the first purpose of this HMP shunt. First, to provide NADPH, and this NADPH will produce or help with the RBC to provide for the antioxidant effects. Okay, and the other thing is that when you in the end by the end of this whole reaction, you produce your ribulose 5 phosphate, and this ribulose 5 phosphate can easily go into nucleotide synthesis. That's very unique about this pathway. It means this pathway can either work as antioxidant pathway. It means this pathway can work as your nucleic acid synthesis pathway, right? And there's another thing, that whenever you have high NADPH levels and you are not having oxidant stress, and it's not your RBC cell, but it's your adrenal cortex, liver, or mammary glands, then this NADPH basically helps with your cholesterol synthesis. Okay, so in your mammary glands and in your liver and your adrenal cortex, this NADPH is converted into cholesterol, like it is utilized into the cholesterol synthesis. That's the main thing. Okay, and of course I told you about nucleotide synthesis pathway. So what are the three main functions of this pathway? It's efficient to provide the antioxidant effects, to provide with the nucleic acid synthesis, and to produce your NADPH to be utilized in your cholesterol biosynthesis. That's the main thing. Okay, and these are like two types of reaction. Your oxidative reaction is the only reaction that produces NADPH. It's the only reaction that produces your NADPH. This can be asked, maybe, that they ask you like what kind of pathway will generate NADPH. Is it oxidative or non-oxidative or something like this? Okay, Your non-oxidative will always only contribute to your either glucose like energy production thing or with your nucleotide synthesis. Okay, and this is present in these cells. I told you their purpose also, so it justifies their presence in these cells. So this is it about this whole disease. Okay, and we can stop here because it's been 1 hour, and then we can continue tomorrow. Okay, and I like if you have, yeah, if someone has any confusions today, we it's like it's too late, so you can like ask me tomorrow and we will start from that. Okay, all right, then I'll see you tomorrow, guys. Bye.